right. Well, I guess we can get started. Looks like everybody is fed and present. And I know James DeGregori always likes us to start on time, everything on time as much as possible to encourage people to actually show up on time. So anyway, it's really my pleasure to introduce Amanda Charlesworth today. I first met Amanda, I think we were thinking about 4 or 5 years ago when she first moved to Colorado. She had an interest in RNA binding proteins and translation, which kind of meshed with some of the interests of my lab, and so I got a chance to hear about her research then, and obviously it's evolved a fair amount since then. And she's going to tell us about today, but a little bit about her. She got her PhD at what was then University College London and the Imperial Cancer Research Fund. Now I guess it's just called Cancer Research UK. And then she came across the pond, was a postdoctoral fellow in the Department of Molecular Oncology at Bristol Myers Squibb in Princeton. Then went to the University of Chicago as a research associate, then a research assistant professor at the University of Arkansas. So she's maybe lived more places in the U.S. than many Americans have. But while she was there, she served as the director of the Molecular Biology Core Facility. She also was awarded an American Cancer Society Scholar Award. And then in 2010 moved to Colorado, and we were fortunate enough now to have her at the University of Colorado Denver at the downtown campus in the Department of Integrative Biology, where she's continuing her interest in RNA binding proteins and cancer. And so, as you might imagine, at the downtown campus, she is busy teaching. She told me today she's teaching 2 classes right now, 2 courses, but she's on sabbatical right now, which means she can think about science maybe a little bit more directly and come give talks to us, and so that's great. So she's also continues to be active in the American Cancer Society. In the last few years, she's presented at a number, I think 1 or 2 Relay for Life events. And she's got some really nice publications, including one that was just accepted and should be coming out very soon, I guess, right? The date was 2017, and I think that's some of the data she's going to tell us today.
Friday was its official publication date.
All right, so there you go. So welcome.
Thank you. Thank you very much. Thank you for having me. So, as you've just heard, I spend most of my time these days lecturing to undergraduates. So I might lapse into undergraduate mode, and if I do, I apologize for that. It's a while since I've done a proper research seminar. So I don't have to tell you this, but that's the view from my lab downtown. So I get to look at a Sports Authority field every day in the mountains, just like you do. So I'm going to tell you about these RNA binding proteins called zygote arrest, or Zar. And because this is the cancer series, I thought I'd start by giving you the cancer angle first, and then telling you what I do. So there's been these observations in the past few years, from about 2010, that Zar has got this strange methylation pattern in some cancers. So here's the first study that showed this. What they were doing was they were inducing tumors in mice, and then in mouse skin, and then they took the mouse skin and the tumors, and they looked for areas of differential methylation. They scanned the entire genome for areas of CpG methylation. And what they found was hypermethylation of exon 1 of a gene called Zar1. And they went into some human melanoma samples, and they found in those melanoma samples that exon 1 of Zar1 was also hypermethylated. And this is kind of weird, because normally hypermethylation in cancer is on promoters, and it results in repression of gene transcription. But this is hypermethylation of exon 1. Now, mention what that might do in a minute. But then following that study, this group continued looking at other cancers and found that more and more cancers could be distinguished. They could be almost diagnosed by looking at methylation of exon 1 of Zar. So, for example, some brain tumors, things like astrocytomas, glioblastomas, oligodendrogliomas, adenomas. All of those cancers has hypermethylation of exon 1 of Zar1. But not all brain cancers. They looked at some brain cancers where Zar was not methylated. And also neuroblastomas as well. Later, they were looking, it was the same group, they looked at hepatocellular carcinomas and found the same hypermethylation of exon 1 of Zar1 there as well. But when they looked in bladder cancers, it was different. It was still aberrant methylation, but this time it was hypomethylation rather than hypermethylation. So something weird is going on. But it looks like there's a correlation between methylation of Zar1 gene and cancers. Now, a different group took a different approach to looking for genome methylation. And this time, their starting material was cervical cancer. And they used a microarray setup to look at differential methylation. And they found hypermethylation of the promoter. Not exon 1, but the promoter. So, in a variety of cancers, there seems to be a correlation between methylation of the Zar1 gene and cancer. And in fact, some of them have correlated the amount of methylation with progression of the cancer as well. So it could be a useful diagnostic marker. So what might this methylation be doing? Some of these cancers were shown to express Zar1. Zar1 is highly restricted to ovaries. Normal Zar1 expression is highly restricted to ovaries and mostly oocytes in ovaries. And so there was this ectopic expression in some cancers. So it was the melanomas and the neuroblastomas that were also ectopically expressing Zar. So Zar is normally expressed in oocytes. And other groups are using Zar1 as a different type of marker. So some studies, and it's actually mostly livestock studies, they're using Zar1 because it's highly expressed, it's very restricted in its expression to oocytes. It's been used as a marker of developmental competence. And so these studies are looking at aging oocytes, and they're looking at Zar1 to see if they can correlate developmental competence with aging using Zar1 as a marker. In other studies that were trying to figure out how best to freeze oocytes, these studies were using Zar1 as a marker of developmental competence after freezing of immature oocytes. And then, and I think this comes from CSU, they have a livestock herd and some of that livestock, a beef livestock, some of them have an androgen excess, which seems to affect fertility. They're using Zar1 as a marker for developmental competence. So this is a really strange gene. The methylation of it is thought, people are thinking about using it as a marker for cancer. Other people are using its expression as a marker for developmental competence. So what is it, and what does it actually do? So Zar1, its full name is zygote arrest one. It was discovered in a screen for maternal effect genes, and it was discovered back in 2003. Female mice that are null for Zar1 are infertile. Very few embryos reach the 2-cell stage, and none of them reach the 4-cell stage. And again, I'm going to remind you that its expression is confined to oocytes. But that's not the end of the story. There's also a Zar2. And this is mostly where I come in. I found Zar2 in a screen when I was looking for RNA binding proteins. And so what you're looking at here is an alignment that we published a few years ago. And this is, back in 2013, we scanned the databases and we found every Zar sequence we could find. And we found that, and so these are all the species here, which of course you can't read, but they're all vertebrates. There are no invertebrates in there. And if you use your imagination, you might be able to see some Zar-like sequences coming in chordates. But the homology is not as good in chordates. They're very strange proteins. They're entirely novel. They don't contain any recognizable protein domains or motifs. We're working in the dark here. We don't know how to manipulate these proteins. So what we did, we found all the sequences we could and we aligned them. And here, this is the N-terminal of the sequence, and then this is the middle bit of the protein, and then this is the C-terminal part of the sequence down here. And hopefully you can see some shadowing. So in the N-terminal, there's a block of homology in the N-terminal. And we found out that that regulates mRNA translation, and I'll show you some of that in a minute. And then in the C-terminus, there's also another block of homology. And I'm going to show you that that C-terminal binds RNA. We found that there were 12 invariable cysteines, and I tried to mark those in yellow. Don't know if you can see yellow stripes, but those are 12 invariable cysteines. 8 of those are found in pairs. So there's 2 pairs of 4 cysteines, which is a bit reminiscent of a zinc finger, but not like any zinc fingers that have been characterized. What we found was that all these sequences fell into just 2 categories. There was Zar1, and we called those Zar1 sequences and Zar2 sequences. And we found that these could be designated by these conserved amino acids in the C-terminus. So wherever you see green stripes, those are amino acids that are specific to Zar1. And wherever you see pink stripes, those are amino acids specific to Zar2. So we found that all these sequences fell into just 2 groups, Zar1 and Zar2. Now, turns out that fish are weird, and fish have 4 Zars. Some fish have up to 4 Zars, and they can't easily be characterized into Zar1 or Zar2. But amphibians, birds, mammals, reptiles, they have Zar1 and Zar2. So just so Zar2 doesn't feel left out, there have been some implications of Zar2 in cancer as well. So rather than the Zar2 gene being methylated, this time Zar2 has been implicated in regulating the expression of the BRCA2 gene in breast cancer cells. And it's also important in development. Again, its expression is highly confined to oocytes, just like Zar1. And one study here expressed a truncated form of the protein and got embryos, mouse embryos, to arrest at the 2-cell stage. And this study over here ectopically expressed Zar in an 8-cell Xenopus embryo and got loss of neural tissue and promotion of epidermal tissue instead. So it does something in development as well. But we still don't know what these proteins really do. So this is where I come in. This is what I'm trying to figure out. What is the molecular mechanism of Zar1 and Zar2? For my studies, I've been using Xenopus, the African clawed toad, Xenopus laevis. We use them because their oocytes are nice and big. So these are single cells, and they're 1 millimeter in diameter. They're huge. A single cell is 1 millimeter in diameter. That means it's really easy for us to inject anything we want into them to manipulate their properties. They've also got this really neat trick where they tell us what stage of the cell cycle they're at. So immature oocytes are in prophase of meiosis, and when they get to metaphase, they get these white spots in the brown hemisphere, and this is where the germinal vesicle, or the nuclear envelope, has broken down. And then at metaphase 2, and you probably can't see this, but if you zoom in, there's a little white dot inside the white spot. And that says they're at meiosis, or metaphase of meiosis 2. So let me just briefly orient you to early development of Xenopus. A lot of these concepts are the same in all vertebrate and actually invertebrate development, but the details may be a little different. This is Xenopus details. So here's oogenesis. So oocytes are arrested in diplotin of prophase of meiosis 1. Cell cycle arrest. Here they grow in size, and you can see the little stage 1 oocyte, and it gets big to a stage 6 oocyte. At that point, when it's fully grown, that oocyte then remains dormant. It remains dormant while it waits for ovulation. It can remain dormant for days, weeks, or even months. Possibly years. Xenopus have a lifespan of a few years, so they can probably go a few years without actually, with these oocytes remaining in prophase of meiosis 1. To get them ready for fertilization or ovulation, they re-enter the cell cycle, and we can do that in the lab by addition of progesterone. When we add progesterone, they re-enter the cell cycle. The germinal vesicle breaks down, that little white spot appears on the animal pole, and that's metaphase of meiosis 1. They continue through metaphase of meiosis 2, where they arrest again. And once they reach metaphase of meiosis 2, they're now a mature oocyte, or an unfertilized egg. And this process here is called oocyte maturation. Immature oocyte to mature oocyte. At this point, they can be fertilized, and that 1-cell embryo, or the zygote, then starts embryogenesis. Here's a 2-cell embryo, and this continues to the mid-blastular transition, gastrulation, neurulation, etc. Now, what I want you to note about early development is that while there's lots of transcription during oogenesis, so there's lots of transcription here during oogenesis, and of course that's what makes the oocyte grow in size, and there's lots of transcription for gastrulation, the majority of early development, oocyte maturation and those very first embryonic cleavages and axis formations, they take place in the absence of transcription. There's no transcription. And yet there's differential gene expression. And all the gene expression occurs through mRNA translation. Here are some of the mRNAs we know about. And so some mRNAs are activated shortly after progesterone's added and before metaphase of meiosis 1. There's another group of mRNAs that are activated after metaphase of meiosis 1. And there's another group of mRNAs that are activated in embryogenesis. I've highlighted these three RNAs, Mos, cyclin B1 and WE1, because you'll see those on several occasions today. So different RNAs are translated at different times. How does this occur? How are mRNAs translated at different times? So mRNAs have different combinations of cis elements in their 3' untranslated regions. So this is a messenger RNA, 5' UTR, coding region, 3' UTR. This particular mRNA has 4 different cis elements, all different colors. I've represented them by different colors. Now, in contrast, this is WE, so the top one was Mos, this middle one is WE1. This has a different combination of cis elements, and the bottom one here, cyclin B1, a different combination, again, of cis elements. These cis elements all have specific binding proteins, specific transacting factors, and so all the different cis elements bind the different proteins. So each mRNA is bound by different combinations of proteins. Now, one of these cis elements, I was involved in the discovery of this red one. We called that the translational control sequence. And we found this in both the Mos and the WE mRNAs. And so when I tried to find the protein that bound to this sequence, I found Zar proteins. I found zygote arrest proteins bound to these RNAs. So then what we did was we showed this a few years ago. We showed some in vitro experiments of how Zar1 binds to these TCS sequences. So here is the WE1 3' untranslated region. There are 2 TCS sequences in it, and I've marked those with the red ovals. We labeled this probe, and then we mixed this probe with Zar1 protein, purified Zar1 protein, and then ran it out on a gel. And this is an electrophoretic mobility shift assay, an EMSA, or a gel retardation assay. So here's the free probe. So this probe did not bind any protein. And this probe here has been retarded. Its mobility has shifted. So this is the probe that binds the protein, and this has increased in amounts of protein. So here is Zar1 binding to the wild type we UTR. And then this is where we mutated one of the TCSs and we still got binding. We mutated the other TCS and still got binding. But when we mutate both TCSs, then binding is reduced. So Zar1 binds the TCSs in the we UTR. We also showed that for Zar2, similar experiment. Here's the we UTR, the two TCSs. Here is purified Zar2 binding the wild type UTR. Here, one TCS is mutated, they're still binding. Two TCSs are mutated, they're still binding. Both are mutated, binding is now reduced. So Zar2 also binds the TCS in the we UTR. These are the C-terminal domains of the Zar protein. So the C-terminal domain binds RNA, and we published this a few years ago. So now I'm showing you this again just to remind you of this bit right here. They don't contain any recognizable domains. But they do have these invariable cysteines. And some of them appear in pairs. And so what we did was we mutated some of these cysteines. And that's noted by these arrows here. And so this is purified Zar1 with individual cysteine mutations right here. And any of those cysteine mutations disrupted binding to the we probe. So those cysteines are important for RNA binding. And then what we did was we removed zinc from the binding assay. And in the absence of zinc, the binding is much reduced. So we have pairs of cysteines that bind RNA in a zinc-dependent manner. So this might be a new type of zinc finger. Very similar results for Zar2. We mutated the same cysteines, removed the binding. We removed zinc, reduced the binding. So again, it looks like they're behaving pretty similar. They have the same type of zinc finger. So far, everything's looked exactly the same. Is there a difference between Zar1 and Zar2? So what we did was we did a titration of Zar1 and Zar2 on the we UTR. On the we UTR. So what you'll note down here is that perhaps there is less Zar1 than Zar2 if we look at the western blot of this EMSA, but there is more probe that binds to less Zar1 and less probe that binds to more Zar2. So Zar1 appears to have higher affinity for RNA than Zar2 does. Now, what we also did was, and this was Cassandra Cotter. I don't know if any of you know her. I know that she's somewhere on this campus. She decided that she wanted to mutate those 12 conserved amino acids that distinguish Zar1 from Zar2. And she wanted to see if she could change the characteristics of binding. And I said, you know, that's an awful big effort, and there's 100 reasons why that won't work. But only one reason it will work. So that was good enough for her, and she went ahead and did this. And it actually worked out pretty well. So she took Zar2, which is the low affinity binding. She mutated 12 amino acids, so they look like Zar1. And she actually increased the affinity of that mutant Zar2 for RNA. So it looks like those 12 amino acids that very specifically distinguish Zar1 from Zar2 might be involved in specific recognition of RNAs. So that was some in vitro studies, and we published those a few years ago, and this is the new stuff. So what we've been trying to do since is to show this interaction in endogenous proteins, endogenous RNAs in oocytes. So what we did was we got some GST fusion proteins. We expressed those in immature oocytes. We lysed those oocytes. We affinity purified the GST proteins, extracted the RNA from them, and then analyzed those RNAs by RT-PCR. So at the top here, this is just a Western blot showing that we've expressed GST Zar1, GST Zar2, and then because GST alone is much smaller in size, we actually cut a big white band out the middle of the gel and compress the image a little bit. So if we look at Moz and we, and I've already shown you Moz and we in those cartoons earlier, then you'll see that GST Zar1 and GST Zar2, but not GST alone, will purify Moz RNA. And the same with WE1, GST Zar1, GST Zar2, but not GST alone, will co-purify with WE1 RNA. Now, what I did here was I looked in the databases for some RNAs that had TCS-like sequences in the 3' UTR. And I found these two mRNAs, Physi-related 1 and Pu5, and I'll talk about what they do in a few minutes. But again, it's a similar pattern. These RNAs co-purify with the Zars better than the GST alone. So this is an illustration that the Zar1 and the Zar2 are forming complexes with these RNAs, endogenous RNAs, in oocytes. Then what we did was we wanted to know if the endogenous Zar2 binds. So we developed antibodies to the endogenous Zar2. So in this case, it's just lyse oocytes, immunoprecipitate Zar2, then extract the RNA and analyze it. And so this is without the antibody, with the antibody, and so you can see Zar2 is coming down with the antibody. And hopefully you can see that those RNAs, Physi-related, Pu5, Moz, and We, they're all enriched in the presence of the immunoprecipitated Zar2. So endogenous Zar2 is interacting with these endogenous RNAs. So up to now, the in vitro experiments did show a direct interaction between RNA and protein. But these in vivo interactions are interactions. They could be giant complexes. So next we wanted to know whether there was a direct interaction in oocytes. So we used UV cross-linking. And what this does is it forms a covalent bond between protein and RNA if they're in very, very close proximity, if they are directly binding. So this is without ultraviolet, this is with ultraviolet. And hopefully you can see that in the presence of ultraviolet cross-linking, there is an enrichment for these 4 RNAs, Physi-related, Pu5, Moz, and We. Protein phosphatase inhibitor down here, this mRNA is a negative specificity control. Now, because these cross-linked immunoprecipitates were washed in high salt buffer, high salt, high detergent, any remaining interactions are likely to be those covalent interactions. And so this experiment allows us to say that Zar2 is directly binding these RNAs in oocytes. So, what are those RNAs that Zar is binding? So the first one, MOS. MOS is a MAP kinase. It's required for the re-entry into meiosis. It's one of the earliest mRNAs to be translated. It's also part of cytostatic factor. Cytostatic factor keeps MAP kinase levels high, which keeps the cell in metaphase 2. So this is the cell cycle arrest, while that unfertilized egg is waiting to be fertilized. So MOS keeps that cell cycle arrest right there. WEE1. WEE1 is a later RNA to be translated, and the first time it's used is in the zygote. So the very first mitotic cell cycle in Xenopus takes 90 minutes. The rest of them take 30 minutes. And it's thought that WEE1 extends that first mitotic cell cycle so that the pronuclei confuse. WEE1 has also been implicated in gastrulation. So the early stages, the cleavage stages of embryogenesis in frogs, all these cell cycles are synchronous. And then they start desynchronizing for gastrulation. And this allows the movements in gastrulation when the gut is formed. And it's thought that WEE1 delays the cell cycle in some of those cells to allow for these cell movements. Physi-related is related to Physi. They are both components of the APC, the anaphase promoting complex. This is the E3 ligase that targets proteins for degradation in metaphase. Physi-related specifically is also known as CDH1. In mouse, it's required for fusion of the pronuclei. I don't know, I haven't seen any studies that describe its translational regulation. I don't know when it is translated. And that would be something that we will be investigating in the future. POO5. So this is an interesting mRNA because in zebrafish, this is the protein that is the first transcription factor. This is a protein that activates the zygotic genome. Also in zebrafish, it was noticed that it's one of the most highly translated mRNAs prior to activation of the zygotic genome. In somatic cells, and especially in mammalian somatic cells, POO5 also goes by the name of OCT4, OCT25, several different names. But it's one of the stem cell factors. It's a transcription factor that's important in stemness of stem cells. So perhaps in Xenopus embryos, the Pu5 mRNA is regulated somewhere in embryogenesis. And this is something that we're interested in looking at in the future. When is this translated in Xenopus? So you're probably wondering when I'm going to show you heat maps and RNA sequence data and genome reads. Tune in next year. Hopefully I'll have all that data next year. We do desperately want to know what other mRNAs Zar binds to. We know 4 of them. I suspect there's a couple of hundred that Zar regulates. And so we're well on the way. We know how to purify Zar interacting RNAs. We just need to get that right balance of specificity and sensitivity and then get them out here to be sequenced. So we're working on that. And then once we've identified those RNAs, we want to show a role for Zar in translation of those RNAs. And this is what's been really quite difficult about this project. So this comes back to the idea that Zars have really, they're novel proteins that have these protein domains that are completely unknown. So whenever we try and manipulate these proteins, we get all sorts of artifacts. So this is kind of slow work. They're also very stable proteins. So when we remove the RNA, the protein remains for days. So we can't knock down these proteins. We've made antibodies, injected antibodies. They don't seem to do anything, so we can't manipulate these proteins with antibodies. So we're trying to make lots of different types of dominant negative mutants to see if we can interfere with endogenous Zar somehow there. So that's what we're working on at the moment. And then that will allow us to ask, how does disruption of Zar affect development? So that's where we're going on the RNA side of things. But now I'm just going to finish with, and I'll briefly go through these and just show you the highlights now of what Zar does when it binds messenger RNA. So I'm going to remind you that the N-terminus represses translation. And we showed this a few years ago. The N-terminus of Zar1 and Zar2 repress translation of mRNA reporters. And to show that, to investigate that, we used this tethered assay. So this is a tethered assay. We have 2 luciferases, a firefly luciferase and a ranilla luciferase that's just a loading control. The firefly luciferase has some viral stem loops in the 3' UTR. They're from the MS2 virus. The MS2 virus has this coat protein that binds to those stem loops. And then what we can do is we can fuse our protein of interest onto the MS2 and ask if this protein can affect translation in any way. So we fuse MS2 to our proteins of interest. And we were able to show repression for the N-terminal of Zar1 and Zar2. But we had real trouble showing repression for the full length Zar1 and Zar2. And it turns out that they were highly unstable. And so we had to become protein engineers. And this is what we just had published in Protein Engineering. We had to add linkers between the fusion domains and our Zar1s to make them more stable and to improve their bioactivity. And so these are the constructs that we used. We used an AP linker. This is an alanine-proline linker. It's rigid, and so it pushes the two domains apart. We used this alpha helical linker. This also has some glutamates and lysines, so some charged residues. So again, a rigid helix, it pushes the domains apart, but this time it's quite hydrophilic. And then a GS linker, this is glycine and serine, small amino acids, no structure, so that will be a very flexible linker. And so we used all of these different linkers, AP, EK, and GS, and we measured translational repression from each of these different constructs. So this is now translational repression. And we can get about 50% translational repression, which is a decent, healthy amount of repression in these assays. This is a dose response, and they were all pretty similar. But we decided to use the AP linker in Zar1 and the AP linker in Zar2 to do the rest of our studies with. So let me just briefly do the how Zars might repress translation. So I'm going to remind you of the closed loop model of translation. So this is where the poly-A tail binds to poly-A binding protein at the 3' end, whereas the cap binding protein binds to the cap at the 5' end. And these two are brought together. All the interactions are strengthened by EIF4G. Now, this illustrates an important role for the poly-A tail. And it's this that's regulated in immature oocytes. So here's a maternal RNA in immature oocytes, and I'm using cyclin B1. So the cyclin B1 mRNA is transcribed in the nucleus. It acquires a poly-A tail. It's translocated to the cytoplasm where it is de-adenylated. So instead of having a tail of 100 adenyl residues, it now has a tail of about 20 to 30. And it's also translationally repressed. And then in mature oocytes, the tail is lengthened. It gets about 100 adenyls added. So it is polyadenylated, and it's now translationally activated. Housekeeping genes are exactly the opposite. They start with long poly-A tails in immature oocytes, and in mature oocytes, these tails are shortened, and they're poorly translated. So the housekeepings are the opposite of the maternally regulated. And this is important because different translational mechanisms make use of the different poly-A tails. So some mechanisms will actually recruit deadenylases, and you can see that tail shortening. Some mechanisms don't require a tail at all, and they can repress translation even if that RNA has no A's. Some require a tail of 100 A's to repress translation. Some mechanisms are overridden by a long tail, and some are not. And I just want to point out that this short poly-A tail of about 30 is significant because this allows the binding of about one poly-A binding protein, or EPAB, in oocytes, embryonic poly-A binding protein. So we used a whole bunch of different tails on our luciferase reporters to figure out what the Zars were doing. And to cut a long story short, The black bars are translation in immature oocytes. The white bars are translation in mature oocytes. The first two bars are MS2 coat protein alone, and then this is when the coat protein is fused to Zar1, and this is when the coat protein is fused to Zar2. So when there's a tail of about 33 A's, there's translational repression in immature oocytes, which is relieved in mature oocytes. And this is the same with this construct here. There's repression in immature oocytes and relief of repression in mature oocytes. Now, the difference between these constructs is this C17. This prevents poly-A tail dynamics. It can neither be polyadenylated and it can't be deadenylated. But it turns out that doesn't matter. Zar behaves exactly the same no matter what the tail does. And we can see that down here when we measured the length of the tail. Here, the length of the tail does not change size, to be expected. And here, you can see that tail getting shorter in progesterone-stimulated oocytes, just like any housekeeping RNA. And Zar isn't preventing that. Now, over here on the A100, there are... you can see the immature right here. That tail is still adenylated. So Zars are not recruiting deadenylases to shorten the poly-A tails. So we concluded from this that Zars need a short tail to do their thing, to repress translation in immature oocytes and have that repression relieved in mature oocytes. What we're doing at the moment, and this is what I got carried away with this morning, is trying to figure out what mRNAs are binding to Zar to control this translational regulation. So what we did over here was we expressed GST Zar1 in oocytes, purified it, ran out the complex on a gel, Kumasi stained, cut out the bands, and sent those bands for mass spec. And we got some proteins back, translational regulation proteins. Last week, I finished repeating that experiment, and this time for Zar2 and in the presence of progesterone. And this morning I got these results back, so I have 264 proteins to analyze this afternoon. So that should be pretty fun. But I want to draw your attention to this band up here that changes in response to progesterone, and this band here that changes in response to progesterone. What we found was that some of those RNAs, some of those interacting proteins were confirmed with co-immunoprecipitation assays. So I just want to say right off, we didn't see anything with 4G, and that's to be expected because usually 4G is removed for translational repression. But we saw a differential interaction with 4ET and an differential interaction with a form of the cap binding protein, EIF4E, that's actually pretty bad at binding cap. So we think this is how Zar is repressing translation. We think that it's doing it by incorporating, and one of those was EPAB, right here. It binds EPAB in immature and then not in mature. And that's actually very similar to how CPEB represses translation. So We're going to continue working on this, but I just want to conclude that Zar1 and Zar2 repress translation in immature oocytes, and this repression is relieved in mature oocytes. And this is actually consistent with a need for translational repression during oogenesis. They recruit 4ET and EIF4EB in immature oocytes, again, consistent with a need for translational repression of maternal mRNA during oogenesis. And they require a short poly-A tail. And this is consistent with a model that involves EPAB. So the implications of all of this are that Zar proteins regulate the translation of maternal mRNAs important for the cell cycle and activation of the zygotic genome. And if we wrap it all the way around to this seminar series, which is the Cancer Biology Seminar Series, this might mean that ectopic expression of Zars in certain cancers might lead to the regulation of messenger RNAs that regulate the cell cycle, like WEE1 and Fizzy-related, or differentiation, like POO5. And so at that point, I am going to stop and acknowledge some people, some of who I know are actually in the audience. Hello to Momi, hello to Justin. Thanks for the shared resources on this campus for analysing our things. Thanks to the American Cancer Society for funding. And thank you for staying to the end. I'll take questions. Thank you.
Actually, I have a question. So, can you said you wanted to expand a little bit for the more being bound by Zar. Can you make predictions just based on what you know of the sequence that Zar binds?
So the question is, can I make predictions of what Zar binds to? And that's sort of how I found Fizzy-related and POO5. So we knew that Zar might play a role in the embryonic cell cycle, that very first embryonic division from the mouse studies. So I looked for mRNAs that were important in that first mitotic cell cycle, and I looked to see if any of them had TCSs. And then I did the same thing with activation of the zygotic genome. So there's POO5, there's NANOG, and there's SOXB, but only POO5 has a TCS, and NANOG and SOXB don't. So I half did that, yes.
Correlate with the expression level of Zar?
Does the methylation of the gene observed in cancers correlate with expression of Zar1? Sometimes. So in melanomas and neuroblastomas, yes. In the other brain cancers, no. But they think it's a combination of histone methylations as well as DNA methylations, because the histone methylations on the Zar gene were also strange in those cancers that ectopically express Zar1.
Restricted to oocytes.
Yes. Yes. Yes, Zars are normally restricted to oocytes, yes.
When during follicle formation does the ovary Zars come on, or is it even earlier before you nerve cells?
Oh, that's a really, really good question. I would love to know the answer to that. What I do know from, we looked at stages of oogenesis, and they are all highly, Zar1 and Zar2 are highly expressed in stage 1 oocytes. But we haven't gone any further back to primordial germ cells to see when they actually start expressing. That would be really good, yeah, yeah.
All right, well, let's thank Amanda again.
All right, thank you. Well, Well, I can.